Hello everyone, my name is Siddharth and I'm a research student at the University of Toronto. I also go by Sid. Today I will be speaking on behalf of my teammates shown here, and I will present our framework for building machine learning systems that can interpret chemical agglutination tests. Before I begin, I would like to thank the University of Toronto, the Chai Lab and the Wheeler Lab at the University of Toronto, and the National Research Council of Canada for supporting this research endeavor. To start, I will introduce the idea of rapid diagnostic tests, or RDTs for short. RDTs are cartridges which intake some sort of human biomarkers such as saliva, blood, urine, and engage a chemical reaction to identify a certain type of disease state. For example, RDTs exist for identifying influenza, as shown here, malaria, as shown here, and even simple pregnancy tests can be considered RDTs. RDTs and these point-of-care tests are effective for controlling community epidemics. And the reason why is because they're very inexpensive, they are very convenient, and when the results can be interpreted correctly, they can become pervasive among the communities and lead to actionable outcomes in the individuals. However, interpreting results from an RDT is a lot more challenging than it seems. The visual inspection process depends entirely on a user's visual acuity, which is different for everyone. In addition, many studies in the literature have shown a large variance in RDT interpretation results, implying that users make frequent mistakes and the interpretation process is highly subjective. As a result, as RDT designs become more complex, their usability decreases significantly. In this presentation, we will be focusing on a specific kind of RDT called an agglutination test, or AT for short. Agglutination tests, or ATs, are composed of special beads in a liquid form, and these beads bond to target viral particles. And they form clumps when the bonding has occurred. Such viral particles can be from malaria, influenza, or the COVID-19 virus, and if clumps appear in the agglutination tests, that means that those viral particles were present in the user's biomarker. There's a standardized score from 0 to 4 for agglutination tests, where each level corresponds to the degree of clumping and bonding. For example, we have an agglutination test for COVID-19. On the leftmost image, we don't see any clumping, so we'd give it an agglutination score of 4 and say there's no viral particles. The middle image shows some clumping, but not too much, so we'd give it an agglutination score of 2, and we would still say that there are some viral particles. And the third image shows much more clumping, and so we would give an agglutination score of 4 and say, yes, this has a significant amount of viral particles. Agglutination tests share the same benefits of RDTs, so they're inexpensive and convenient, and they offer many options for the RDT developers on the chemistry side of things. One of them being that they can model more complex diseases. However, as you can see, agglutination tests can create uninterpretable structures which are even more complex than the ones we saw in the other RDTs, and that's what makes them unusable for the general public. So it's very unlikely that a user outside of the lab will be able to interpret these images and arrive at these agglutination scores. Our group asked, how can we make agglutination tests more accessible to the general public? This sparked our research effort, which is to create an open source framework that can help agglutination test developers create software for evaluating and improving the usability of their tests. With our framework, agglutination test developers can create apps for end users that automatically interpret their tests, removing ambiguity in visual inspection and significantly increasing accessibility. The process of how our framework can be used is shown here. It starts with a data collection process, which requires agglutination test developers to take tightly cropped images of their test. Next, they can upload their images to our annotation website and use our annotation features to assign consistent and unbiased labels alongside other annotators. Once they have a robustly labeled dataset, they can move on to our image processing pipeline, which extracts key image features specific to clumps that are seen in agglutination tests. The image processing pipeline creates a feature vector for each image, which will be processed by machine learning models in our modeling environment. These models are evaluated in the next step by a set of post hoc analysis tools, which can evaluate the level of concentration that can be automatically recognized from the agglutination test, and also visualize how the tests evolve over time. These steps are iterated again and again until a machine learning model that is trained makes accurate agglutination score predictions. And as such, this final model can be deployed in a smartphone app that end users can use to automatically interpret their test. We will now describe each step of the model development process and at the same time explain how we apply those steps in 2 case studies. The first study is for an agglutination test for COVID-19 detection, and the second is for an agglutination test for blood typing. As you can see, as the clumps get larger in the COVID-19 test, the agglutination scores increase. We see an analogous idea for the blood typing tests. However, we see a high score for a single small clump, a lower score for scattered clumps, and a score of 0 for just a single blood droplet with no clumps. Our framework is shown to make both of these 8 agglutination test designs more interpretable, even though they model completely different chemical and clinical processes and use agglutination scoring in different ways. So the first step of the development process is data collection. And we really only have 2 constraints for this process. We require that the agglutination test developer collects images that exhibit a full range of agglutination scores. So that a machine learning model has enough labels to learn. So for instance, a machine learning model needs to know what an agglutination score of 0 looks like and what a score of 4 looks like. And our second requirement is that the images collected must be tightly cropped around the agglutination test boundaries. For the COVID-19 agglutination test, a 96 well plates was used, where each well held a single agglutination test and was injected with a different concentration of COVID-19 viral particles. The well plate was inserted into an LED light box, and a smartphone camera would take a picture of the plate. This resulted in a grayscale image of a 96 well plate, and the circles in this image were detected using a Hopf circle detector, and each individual agglutination test image was cropped out, as shown here. For the blood typing agglutination test, photos were taken after placing the tests on a grid-like template. Then image processing algorithms used information about the grid template to tightly crop images around the test. The next part of our pipeline centers around the data annotation, which is an ambiguous process in itself. To highlight ambiguities in data annotation, we can take a look at this figure. The top 4 images show COVID agglutination tests with an agglutination score of 3, and the bottom show agglutination tests with an agglutination score of 4. Now, as you can see, the decision boundary between 2 and 3 in this example is quite subtle, and it's very easy for a human annotator to mislabel images in both of these classes. If there are multiple annotators, it's also easy for some annotators to label more 3s than 2s, or vice versa, and that creates inconsistency in the labels. We built a web annotation framework to solve this problem and help facilitate labeling datasets so that they're as denoised and consistent as possible. In the center of the website, one image is shown at a time in a randomized way so that we mitigate potential anchoring bias. The bottom left-hand corner shows a formal criteria for what each decision for assigning agglutination scores should look like. These are uploaded by a lead investigator and should comply with the standard agglutination score criteria. The right side of the interface shows that a list of prototypical examples that an annotator can use for side-by-side comparisons. If they're unsure if an image is a 2 or a 3, they can cycle through those examples. And these reference images are also uploaded by a lead investigator. Finally, annotators can flag images for which they're uncertain about the label, and those images are escalated to other annotators for review so that everyone has a chance to label that. All of the features on this interface are entirely configurable through a single config file, so no web programming experience is required to customize it. As an example, we just modified that previous config file, and now we have a custom web interface for the blood typing agglutination tests. Our framework also provides scripts for analyzing annotations post hoc. Confusion matrices for the labels between all pairs of annotators are generated to see at a glance the overall agreement in labels, and also see if anyone is over-annotating or under-annotating. We also visualize in our scripts the specific images that have high disagreement so that annotators can meet and sync on those images to learn from each other. The second component of our framework is a computer vision pipeline for automatically calculating image features that characterize the degree of agglutination in an agglutination test. So we see three images. The first one are images of the COVID-19 agglutination test as is. We then apply brightness and contrast adaptive histogram equalization, and specifically we use an algorithm called CLAHE. And then we apply global image thresholding and adaptive image thresholding to create a binary mask that isolates the clumped regions from the background. And we use that mask to generate our image features. Once we have separated clumps and the background, we can compute these following image features, which we've experimented and refined in our case studies. In our framework, it's also very easy to add new image processing functions to different agglutination tests. And adding a new function is as simple as including its Python code and adding it to a configuration file. In addition, hyperparameters can be customized using a configuration file. So in our case studies, we had the same code for the COVID case, and we just changed some hyperparameters to apply it to the blood agglutination case, and that worked out just fine. Now that we have a labeled data set of image feature vectors, we can start training machine learning models in our modeling environment. We use cross-validation sets in the training process and allowed for agglutination test images to be part of groups such that they won't separate alongside the cross-validation sets. An example of a group can be seen here. So if we look at the first image, this is an agglutination test, and we see that it's at time 55. And we have the same agglutination test at time 60. Now, we don't want this image to be in train and this image to be in test. And so our cross-validation groups prevent that from happening. From there, training is just a matter of feeding in the data set of feature vectors with its label to a machine learning model. We tried out various machine learning models: linear regression, logistic regression, boosting trees, AdaBoost, and we found that gradient boosting trees work best. Because agglutination scores are ordered, all of our models were regression models, which rounded to the nearest integer as a final agglutination score prediction. Finally, our framework presents a set of post-hoc analysis tools that can be used to analyze the sensitivity of the automated agglutination interpretations. An example of the post-analysis tools is shown here. For the COVID-19 agglutination tests, we can see a plot of agglutination scores over time, and this shows with each line corresponding to a concentration of the virus, and it shows how each concentration evolves its agglutination score over time. And we can also see some separability between the high concentrations and low concentrations. The image on the right determines this separability using hypothesis testing at each point. So as you can see, at time t equals 72 minutes, the agglutination tests with concentration 120,000 PFU per ml is statistically distinct from the concentrations lines of the other concentrations for this virus. And this type of analysis can provide insight on what concentrations can be detected from the agglutination test. Swarming plots can also be visualized by our tools to see how predictions compare against labels. And for our specific case studies, we realized that most end users really only care about making binary decisions with these agglutination tests. So negative versus positive. And if we use a decision rule where 0 is negative and anything else is positive, we found that the blood agglutination tests had an accuracy of 97%, and the COVID agglutination test had an accuracy of 82%. So finally, when a machine learning model is iterated and trained through these steps, and it's working as intended, it can be deployed in a smartphone application that automates interpretation for users. This last step leads us to future work in the area of rapid diagnostic test development. New SDK libraries can be made for RDT developers to create apps that improve the accessibility of their tests. One such SDK is RDT Scan by Park et al., which guides users to take a good picture of their RDT with appropriate lighting conditions and template matching. Another SDK is introduced by Antiomopoulos et al., and it integrates colorimetric reference rates to aid in model calibration. So if color is an important feature, this SDK can be useful. In addition, if you have a larger data set, you can use deep learning to automate the feature extraction section of the pipeline. And Alongside a larger data set, you can experiment with sequence models, which can utilize information on how reactions evolve over time. Thank you very much for listening to our work and our presentation, and I'm more than happy to take any questions.